We have the pleasure to welcome Professor Ramsey as a keynote speaker for CE Pharm 2025. Professor Ramsey is the Minnie N. Goldby Distinguished Professor of Chemistry at UNC Chapel Hill, where he has built a remarkable career as a pioneer in microfluidics, micro and nanofabrication, analytical chemistry, and bioinstrumentation. He's widely recognized for his groundbreaking work on microchip-based analytical devices, technologies that helped launch the era of lab-on-chip systems. These innovations have transformed chemical and biological analysis, dramatically reducing sample sizes and analysis times, while expanding what is possible in genomics, proteomics, clinical diagnostics, and pharmaceutical development. His research not only advances fundamental sciences, but also bridges the gap between academic discovery and real-world application, reshaping laboratory. Professor Ramsey's contributions have long been a part of CE Pharm, showcased both in the scientific program and in the vendor hall. His pioneering work with calipers microfluidic lab-on-a-chip technology applied to DNA, RNA, and protein analysis is reflected in Revity, LabChip, and Agilent Bioanalyzer. More recently, his innovations have taken shape in 908 devices, a zip chip platform, which couples microfluidic capillary electrophoresis with mass spec for protein, peptides, and amino acid analysis. These are just a few examples of Professor Ramsey's ventures, and each contributes to shape how we think about speed, efficiency, and precision in pharmaceutical sciences. His leadership and vision have earned him numerous accolades, and his impact continues to be felt across our industry. A couple of fun facts about Professor Ramsey. He's not only a great scientist, but has been a big game fisherman. Right. He has exquisite taste in food, always knows the best restaurants wherever you go. Please join me in welcoming Professor Ramsey.
This is our room to advance. Yes, okay. Good morning. Yeah, thank you for the kind introduction, Mohammed. We were talking before the meeting started here, so 2018 was the last time I attended CE Pharm, and I think I was involved in CE Pharm before that as well. So it's really nice to be back, and I very much appreciate the invitation to speak here this morning. So I just want to let everybody know, right, that I'm involved in commercialization and, products from these companies spun out of my laboratory, and I'll be talking about some of that here this morning. So activities, so actually my training originally was in laser spectroscopy and laser technologies. So it was in the late 80s that I got interested in micromachining and microfluidics and started activities in the early 90s. So this device shown on the screen is the first device that we published on that was CE on a chip. I'll talk a little bit more about that in a bit here. That led to products out of Agilent, so Caliper Technologies, commercialized lab-on-a-chip technologies, and we did a co-development agreement with Agilent that launched the Agilent 2100 that maybe some of you are familiar with, and then eventually PerkinElmer and now Revity, right, is now selling those products. So all of those products involved using laser-based detection to detect fluorescent tagged molecules, right, after the CE separation, but then we got interested in doing electrospray so we could actually do further characterization of molecules. And so we integrated monolithically nanoelectrospray onto these microfluidic CE devices. In the early 90s, we also got interested in miniaturizing mass spec, so we had success with microfluidics, so what other analytical tools could we shrink? And so we looked at micromass spec. That work started in 1996, which seems like a long time ago now. I guess it is a long time ago. But anyhow, that IP led to the formation of 908 devices and the handheld mass spectrometry. That was the seminal products launched at 908. And then we brought the CE electrospray devices into 908 as well, called Zip Chip as the product. And then, of course, if you have a mass spectrometer and you have a separation tool that has electrospray, right, you're going to want to marry those. And that led to a product called the Rebel that I'll touch on later a little bit. We also worked in nanofluidics. So this is a picture of a 50 kilobase strand of DNA here that's labeled with an intercalating dye, so we can see it fluorescently. It's moving through a 200 nanometer channel here. And we had a way to turn on a restriction enzyme inside of this nanochannel, so it cuts at specific sites in the DNA. So it's intact at the beginning of the channel on the left-hand side, but by the time it travels to the end, it's actually cut at specific sites for the enzyme that we're utilizing. And we can measure the integrated fluorescence of those peaks, eluting the area of those peaks gives the size of the fragments. And because it's a nanochannel, they can't pass one another, so we also get order. So this was a tool that we tried to develop to look at long-range structural variation, long-range genetic variation, so-called structural variants. So that was a company, Genturi, which, as you may know, some startup companies end up belly up. And so actually I bought all their assets for $3,500, so it's not completely dead yet. And we moved the nanotechnology, the nanofluidics into plastic devices so we can make them relatively inexpensively. And then the last area that we worked on, and I'll talk about that some at the end, it doesn't involve electrophoresis, but involves characterization of proteins and nucleic acids, so hopefully there'll be interest here in that as well. But it's a way to do massively parallel PCR reactions, so digital as well as quantitative PCR and immuno-PCR types of reactions. So this was, say, the first device that we published on, CE on a chip. So yeah, in 1993, I was at Oak Ridge National Laboratory at that time. So yeah, it's like a big number, right? Between now and then, 32 years ago, it's yeah, I always tell people that I actually started on this work when I was a child. So just anyhow, we, these devices, we use photolithography and wet chemical etching to form trenches into a glass substrate. Right, we have 4 reservoirs that are connected to the various termini of those channels. Channels are, in this case, about 75 microns wide and 5 microns deep. And then we have electrodes sticking in each of those circular reservoirs, and electric potential is applied to control transport into the channels. That 4-way intersection we can make act as a valve by applying time-dependent voltages. And yeah, at that first talk in 1993, I tried to go back and find some of the separations that we produced back then. But they were pretty bad, so apparently I threw them all away. I haven't been able to find them, but anyhow, eventually we got better. So this is a 3.5 centimeter separation channel. Again, that 4-way intersection that, again, by applying time-dependent voltages to the various channels, or reservoirs rather, we can inject a very small plug, typically sub-nanoliter in volume, and perform a separation here. Nothing very interesting, dichlorofluorocene and an isoform there that's being separated. But we're generating 25,000 plates per second in this separation. So very rapid performance in terms of separative performance in this device. And then say that the IP we generated there led to the formation of Microtechnologies, renamed Caliper Life Sciences, and acquired by PerkinElmer in 2011. Say there's the Agilent 2100 there in the upper left. So yeah, I learned in preparing for this talk that that's no longer sold as of the end of 2023, but the the chips are still being sold. Yeah, and some heritage products there. The other one that is still alive is the GX2 Touch, sold by Revvity now, right? A divestiture of the life sciences tools and diagnostics products from PerkinElmer into this new entity. So just, I don't think anyone in here probably has seen this slide before, something that we didn't publish on this specific setup, but we had published on a monolithic 2D device to do 2D separations based on hydrophobicity followed by CE, size to charge ratio separations, generating like for a peak capacity of 4,000 in 13 minutes. But that utilized MEKC for the hydrophobic separation, and people who've performed MEKC probably know that it's not a very robust technology. So we moved to this hybrid sort of setup where we use capillary LC, 75 micron LC, half meter long, and coupled it to a chip to perform CE. So we have wide peaks, relatively wide peaks. I think they're like 25 seconds wide coming out of the LC column. And then we can do rapid CE separations as those peaks elute, multiply sampling the peaks as they elute out of the LC column. And I just in that molecule there in the lower left, right? So we're using a dynamic coating here to try to keep peptides from sticking to the surface. And this is sort of data that we could generate. So we're using, again, laser-induced fluorescence. So this is that's sort of far away. That's a BSA tryptic digest in this particular case, right? That is fluorescently labeled with TRITC. So again, as I said, we can use laser-induced fluorescence detection, which is very sensitive. We're generating here, if you're optimistic about using that full 2D space here, over 7,000 peak capacity. You can see as the hydrophobicity increases, right, the charge heterogeneity is reduced. So we're really only using about half of that space. So we say we can generate 2,500 to 4,000 peak capacity in this separation, which is unfolding at LC, cap LC sort of separation times, 70 minutes in this particular case. So fluorescence is really nice, right? Very high sensitivity. You can see in the notes up there, right, those peaks, the base of the peaks for the coming out of the CE are 43 milliseconds wide on average, right? So they're very short, right? And so it's, so that's a nice thing about laser-induced fluorescence detection, but the bad part is I just drew a square around one of those peaks. Like, what is that peptide? We have no idea, right? Laser-induced fluorescence allows us to detect it, but it gives us no chemical information. We have some chemical information about the hydrophobicity based on the LC and the size-to-charge ratio based on the CE migration time. So, that led us to think about integrating this electrospray capability onto chips. So we first published on that a strategy to do that in '97. I think one of the two publications with my wife there. So she had a background in CE electrospray using capillaries and had an interest in integrating it onto chips. So the chip structure is, again, this four-way intersection that's shown on the right-hand side there to do the valving and injection. Then we form this junction in the lower left-hand corner, right, which allows us to make an electrical connection and a fluidic connection there. And then we direct the channel to the corner, right? We want a sharp feature to do electrospray from. So you look at a rectangle, right? The corner is the sharpest feature. And so that's what we do. You can see by the legend there that we've actually coded the separation channels to be positively charged. And then the side channel is just native silica. So when we apply the potentials as shown, right, we have electroosmotic flow in the direction shown by the arrow and convergence at that corner, pushing the separated molecules out to the electrospray emitter. And into the mass spectrometer. So it's nice, right? I mean, you don't have to do any sort of fluidic connections like you would with a pulled emitter. And as people who practice that sort of technique know that a common failure mode would be chipping the glass and plugging the electrospray emitter. So we don't have that problem here. We also find that we have a lot of flexibility in terms of alignment. So, this eventually ended up being a product, the zip chip at 908 and now Repligen, but we don't need to move that chip around the electrospray emitter, that we can fix it in one point, and the optimal position is fairly sloppy in space. So what could we do with that? So, this is some work that is a little more recent, only 20 years ago. Showing, in this case, we've actually covalently put a positive charge on the surface. I'll mention that in the next slide. But again, very high performance separations, I would say. So, generating close to 1 million plates here in 3 minutes for these peptides that we have. 6,000 plates per second, so we've had to slow down the separation so the mass spectrometer could keep up. And so I think, if I remember right, so we're taking data here. This is mass spectral data, right? The previous slide with peptides, right, that was mass spec, not LIF. And here, mass spectra associated with all those peaks as well. So we're running the mass spectrometer at 8 spectra per second in this particular case, and the base width of the peaks are a little over 400 milliseconds in this particular case. So, and these separations are within 10% of the theoretical limit, and if you go into the literature, what we found is that sort of the best separations were about 30% away from the theoretical limit. So this is, I think, about as good as you can get, and that's because of the surface coating. So, right, CE is nice because we don't have to have interaction, right, with a stationary phase, but we do have a surface there, the walls of the channel, that we have to worry about interactions with. So, what we eventually did is we found that we could. We wanted to put a positive charge on the surface of the channels, right, because we're interested in separating peptides and proteins at low pH, so they're positively charged. So we want to repel off of that surface, right? The silica, right, is going to natively have a negative charge, so protein positively charged molecules are going to stick to it. So that previous slide or previous separation I showed you just had this APS, aminopropylsilane coating on the surface, and we found that we could get a much more homogeneous coating by using a chemical vapor deposition approach, right? Gas phase deposition versus traditional sort of liquid phase coating to those surfaces. So, and we found that, that works pretty well in capillaries as well. You're limited to the length, right, that you can get the molecules from the gas phase into the channels, but it works quite well for the sort of separation channels that we're utilizing, that typically maximum sort of at 40 centimeters or so. And then when we found that when we went to separating proteins, intact proteins, that we also put this PEG coating onto the surface, so covalently attaching the PEG onto the aminopropylsilane surface. So that would be done in the liquid phase in that particular case. But so fooling around and doing intact protein separations, right, we eventually found that we could do a pretty interesting separation here and intact characterization of an intact monoclonal antibody. So we get charge state variation here in the CE separation. So this is infliximab, Remicade, monoclonal antibody that's off the shelf, right, sold as I think maybe one of the first monoclonal antibody therapeutics. I don't know if that's right or not, but I think it was. Anyhow, we could get these separation of the primary components here, right? Infliximab, where we have N-terminal lysines that are truncated to different degrees, either not truncated at all for the 0K or one loss of or two losses here. And we could integrate the mass spectra over those particular peaks and generate the mass spectral data shown over on the right-hand side. Right, so we can get the molecular weight of the particular components here in this monoclonal antibody, right? We can get the glycosylation. We obviously get the mass, and but by looking at the mass shift for the different peaks, we can see what sort of variation, what sort of post-translational modification is taking place. So the mass shift corresponds to the loss of lysine, and then that loss of charge from the lysine truncation, right, should also equal a constant shift in migration time, which we can see in the CE separation. So yeah, room for improvement in terms of separation of the basic and acidic components in that particular case, but you'll see that it did get better. We moved the technology into 908 devices, became the zip chip product, so all the characteristics that I showed you previously, a little different injection strategy that I won't get into, but a four-way cross again, but using pressure rather than electric fields to do the injection in this particular case. You know, we are in the nanoflow regime here. I didn't mention that earlier, but you know, typically 150 nanoliters per minute sort of flow rate for the electrospray. So in two different versions, right? There's the high resolution version of this chip, which is what I think, does it say on there, 46 or I forget, 40 centimeters roughly, and I forget, maybe it's 30 centimeters. And then a high-speed separation, which is 10 centimeters separation channel. And then this was launched initially for the Thermo Orbitraps and eventually also interfaces for Bruker and SciEx mass spectrometers. So auto sampler, right, to pick up samples out of a 96 well plate and feed them into the zip chip box and into the zip chips. And obviously 908 could do these separations of monoclonal antibodies, and we found the team, mostly people from my lab that moved into 908, found that changing the pH of the buffer, you know, from acidic, you know, pH 2.4 or so to near neutral sort of pH allowed much better separation of the basic and acidic components. Again, you know, mass spectra could be integrated over the individual peaks, and we could see again the truncation, the loss of the lysines, and also the glycation patterns. And they could show that you know, it was generally applicable to monoclonal antibodies, just a collection of different monoclonal antibodies here where these charge variations could be observed. And some more recent data that I got from 908 and Repligen now, sort of data taken with zip chip here. So this is a analysis of spent media, right, where they're producing the NIST mAb with NIST CHO cells here. So it's a small, I think it's a 250 milliliter reactor that they're running. And they're grabbing a sample. In this case, I think it was on day 5, just showing they could dilute the sample. You know, another attractive feature, right, of zip chip is basically doing a dilute and shoot sort of sample prep, so very little sample prep and hands-on time there. But they could, again, separate the charge variants and also get the glycation patterns, and looking at both the CE and the mass spectra, determine the post-translational modifications that are taking place, so different amounts of variation that are taking place with the formation of pyroglutamic acid and with the acidic variation, right, as a deamidation. So they followed this over what I believe it's 14 days here, looking at the variation of the different, the abundance of the different charge variations and the variation in the glycoforms that they could monitor. They have the sensitivity here that they could actually collect a sample right after about a half a day here, and then roughly 2 samples a day. Trying to see how the charge variation, how the product is changing as a function of time through the manufacturing process, and trying to present this to people in the bioprocess community to see if this can be useful to them in terms of optimizing the product. And then moving forward, trying to do direct sampling out of a bioreactor. Basically a pump to bring sample out of the bioreactor, do filtration, and place the samples into a well plate, which then would go to zip chip, or eventually, with some hope, that it would go directly to a chip and do electrospray and the mass analysis. And so this potentially could be, in the case of moving to perfusion reactors, I think, a very important sort of development. Okay, just changing slightly here, right? So, we, in my lab, right, we worked on miniaturizing mass spectrometry that I mentioned early in the talk, and we have the zip chip or electrospray device, and if you have a mass spectrometer and a separation device, you're going to want to put them together. And so we did that at UNC, showing that we could analyze bioreactor broth with that sort of technology, and eventually became a product called Rebel at 908 Devices. And this is some data from a qualification, a sample mix that was run through Rebel here that contains, I think, 36 components, all the amino acids, some dipeptides, some vitamins, and some calibration markers here. And so you can see, I need binoculars here to see this, but let's see, what is it? 18 and 19, right? Leucine and isoleucine, right, that we can't separate mass spectrometry. They have the same mass, right, so we can't do mass analysis to separate them, but they're separated by CE. And then, let's see, I think it's, 22 and 23, which I think are proline and, no, it's methionine and threonine, which are overlap in the CE space, but can be easily resolved in the mass spectral mass space. Okay, so this product is primarily directed towards monitoring bioreactor broth, at line, and just some data here showing that the technique is tolerant to various additives that typically go into a bioreactor broth. And again, same features as with the zip chip, that it can do pretty much a dilute and shoot. Maybe a filtration is necessary as well. So very little hands-on time in sample processing. And Repligen recently launched the latest version of Rebel, they call Rebel XT, which hopefully has some various improvements. The sample list has been dropped from about 3 dozen to, I think, 22 different components here, amino acids and a couple of dipeptides. I'm going to move on. So actually my latest entrepreneurial activity is after the divestment of the life sciences tools vertical from 908 to Repligen. A couple of former Ramsey lab people left 908 and didn't move to Repligen, and we formed a new company called Move Analytical, directed towards using zip chip to do metabolomics analysis. So, I haven't done metabolomics analysis myself, but my understanding from working with my colleagues here and actually talking to people in the mass spec community, particularly people that are quite capable in the proteomics space, find that moving into metabolomics can be challenging. Right? So there's sample prep issues, right, and then just sort of the labor actually going through chromatograms and trying to properly assign peaks based on their retention time and mass spectral results. So we basically are trying to simplify the sample processing and also to automate the assignment of peaks to particular metabolites. And we found that is easier to do using electrophoresis. So we have software that we're developing. So it's a combination of reagent kits and software that we're trying to put together to make metabolomics easier here. Basically, you can design a sample list in an Excel spreadsheet, and then you can just drop that spreadsheet on to a component in the user interface, and it loads all of the material into the appropriate slots. Computationally reloads it. It also loads the execution files for zip chip as well as the mass spectrometer. The sample prep is relatively straightforward, right? There's methanol precipitation, there's some internal standards, stabilized tope labeled amino acids, right, to do a retention or migration time index sample to solidify the migration time and help us with assignment. About 12 samples, 12 minutes per sample, running through a 96 well plate, and then automated analysis assignment of peaks with the software. So, oops, see if I can go back here. I'm guessing people who have done CE are well aware that migration time can move around, right, and primarily temperature dependent because of viscosity, right? Electroosmotic flow and electrophoretic mobility are both dependent upon viscosity. So just some data here using some markers, some amino acids here, I believe, right, without any sort of control. You can see in the center sort of frame a run over a well plate here at 4% sort of relative standard deviation migration time. And then if we do index migration time based on roughly a dozen, maybe 17 internal standards here, that we can lock that down to less than 0.5% relative standard deviation. So it's utilizing a notion that what Kovacs put out for GC back in the 1950s, but and it's used in LC, of course, as well, but really very helpful here in CE. And in CE, right, like leucine and isoleucine, I showed you, and alloleucine, right? We can separate all of those in CE, and the order of their migration is not going to change. But in LC, right, you could have a change in retention time. So that makes the automated assignment a bit easier here, and we're trying to utilize that characteristic of CE in this metabolomics analysis. This is some data from Hampus Engstrom, an ASMS poster, so if you want any details, I know he's here and you can talk to him, but basically trying to use this tool metabolomics capability of looking deeper than what one typically would do in terms of just looking at amino acids or lactate, right? This is, again, an example of a continuous fed reactor, right, generating the NISMAB and under a couple of different conditions, different concentrations of glucose being fed to the reactor. And if we look at sort of the typical components that one would monitor, that you would see that those two different conditions don't show any significant variation. But looking into the glutathione pathway here, a couple of components there that we can see that there's a difference in those two different operating conditions. And so we're hoping that if by digging deeper into the broth, biochemistry into the broth, that can be useful to the, biomanufacturing people. And yeah, this is sort of showing what our capabilities are compared to a couple of popular paths for doing metabolomics there. So advantages in terms of time and cost, and so we think it's something that we're hoping that the community interested in metabolomics will grab a hold of. So it's reagents and software that we're producing here. So I'm going to move away from CE here, and so in the last 10 to 12 years, I've been focusing on something different here. So, excuse me. Actually, this work started in 2011 in my lab at UNC, and so we're trying to develop a platform to allow state-of-the-art sort of sensitivity in terms of detection of proteins and nucleic acids. We'd like to do that at as high a plex as possible, much higher plex than traditional sort of quantitative PCR allows, or immuno-PCR. And we're doing that by doing bead-based types of chemistry. So we're taking polystyrene microbeads and we're tethering either primer pairs to the surface for nucleic acid analysis or capture proteins for the case of characterizing proteins. And so we are taking beads, right, we're coloring them similar to Luminex, if you're familiar with that. So we're encoding them with fluorescent tags at different concentrations, tethering chemistries to the surface, then mixing them all together in an ensemble that are incubated with the sample. So we're using microfluidics here as a way to manipulate these beads and carry out the bioassay, and as well as building the readers. So this is work that was supported by DARPA and DITRA, say starting in 2011. So I think most everyone in here will be familiar with PCR and ELISA, right? They're sort of the workhorses, if you will, for analyzing nucleic acids, DNA, RNA, and as well as proteins, right? They have great characteristics in general in terms of sensitivity and the ability to detect pretty much any sort of reasonable protein or any nucleic acid that we want. Sort of the weak points have been the ability to multiplex in quantitative PCR, and the issue of taking and forming a new panel, right? Typically, if you're going to do a multiplex PCR reaction with a reaction all containing in one pot, you have to work pretty hard to make sure the different primer pairs for different targets don't interact with one another and form what's called primer dimers, giving you false positives. And then on the side of ELISAs, when you start trying to multiplex ELISAs, when you increase the plex, you're adding more and more detection antibodies. It turns out that the noise floor increases, the background increases, and reduces detection limits. And so we think that we have solved those problems. And I'll try to show you that. So this is the assay strategy. So we start with a colored bead, right? And we put primer pairs onto the surface of that bead. In this case, through a thermally labile bond, right? So eventually we want to be able to release these primer pairs from the bead. And the beads are paramagnetic, so we can manipulate them. So different colored beads, different primer pairs on the beads, they all interact with the sample simultaneously. The primer pairs act as hybridization probes to pull the targets out of the sample. Again, beads are magnetic, so we can move them away from the sample, leaving PCR inhibitors behind. At that next center frame there with the white background, right, if we want to do RNA, right, that's where we do the RT step there. And then those beads are magnetically moved into microwells that will only accept a single bead. So we now bring in a PCR master mix sans the primer pairs, because primer pairs are delivered by the beads, right? And we seal those wells with immiscible oil, so there's no communication between the wells. And then we can thermocycle. There's also an intercalating dye that is in there, so that'll give us a readout of the amount of amplicon that we're generating. As I mentioned earlier, when we do that first thermocycle, the primer pairs are released from the surface of the bead, so we have a homogeneous reaction taking place, PCR reaction. And in about 15 to 18 thermocycles, we can detect a single molecule in one of those wells. And we can also, because we monitor some subset, so it turns out, I'll show you that there's 250,000 of these wells in our current devices, so we're doing lots of these experiments simultaneously. So we can detect single molecules in a well. We can also, by monitoring some of the wells, detect 1,000 molecules in one of the wells by qPCR, right? Determining when a cycle threshold is exceeded. And then what about proteins? So we are using immuno-PCR. So we have a PCR platform, right? So we convert the ELISA experiment to a PCR experiment. So we have capture antibodies on the surface of the bead. We capture our antigen. We bring in a secondary antibody that recognizes that same antigen, and that secondary antibody has a reporter nucleic acid attached to it, covalently attached to it. And that reporter nucleic acid is unique to the protein. Right, so we put the forward primer for that unique reporter on the surface of the bead, and then the reverse primer is generic that is delivered in the PCR mix. So again, right, different colored beads, different capture antibodies, incubated all simultaneously. We remove the remove the beads from the sample matrix. So now we can bring in our secondary antibody here in the next step. And then the beads are delivered to individual wells. PCR reactions take place. Again, we can detect a single protein, single molecule in one of those wells, as well as multiple 100 or 1,000 by digital PCR for low concentrations and qPCR for high concentrations. I'll get to that in a minute. Okay, so we're turning off nonspecific binding here by using this unique nucleic acid reporter in these experiments. So in the case of the blue antigen and the beads with the blue capture antibodies, right? So in that case, we produce a fluorescent signal when we do our PCR amplification because we have the forward primer from the bead and the reverse primer from the solution. But if a red antibody or a green antibody non-specifically binds to the blue beads, we don't have any amplification because we don't have the appropriate forward primer to amplify that nucleic acid reporter. Okay, so I said we could detect a single molecule in a well, so we're doing digital PCR, right? So in digital assays, you take your sample and you divide it up into lots of small components or partitions, right? And by reducing the volume in those partitions compared to the entire sample, you have low enough background that you can detect a single molecule. And so at low concentration conditions, you have 10,000 partitions, right? You can count how many of those partitions give you a positive signal, and that will allow you to directly calculate a concentration. Right? It's Poisson statistics at that point. Eventually, as concentration increases, as we move from left to right, all the wells become positive. So in the digital space, you lose your ability to do quantification. But at that point, we can use our qPCR data, right? Because we're monitoring some fraction of the wells after every cycle, and we can still do quantification. So we can end up getting like 8 logs of dynamic range in this experiment. Just very quickly, I said we're using beads of different color. If you're used to flow cytometry and scatter plots, right? We can we're using 2 different wavelengths in this particular case, and we can measure the intensity at these 2 different fluorescence wavelengths and decide, which one of these sort of areas that bead falls into, and that tells us what target is in that particular well. And here we're going to do a little experiment. So this was one of our early chips. It only has 40,000 wells in it, and so the wells are not just cylindrical sort of pits, as you can see that they have this keyhole sort of shape. So if we look at the micrograph, right, you can see the keyhole shaped sort of well. So those the small dimension, the width of the tail is about 2.5 microns, and the head is closer to 4, and they're 4 microns deep. If we look at the fluorescence micrograph all the way to the right, you can see that the beads all light up because the intercalating dye partitions into the bead, right? So that tells us if there's more than one bead in that well, right? So that actually is an analytical tool for us. But you can see that there's some of the tails light up, and it's only when the tails are bright that you have a positive signal. So watch closely and you'll see these tails grow in, right? So these are snapshots after each PCR cycle, right? So we can count up the wells that have positive tails, so that's a positive signal. And we can look at one fluorescence encoding wavelength, right, and look at the intensity and look at a second one, and by looking at those two, we can decide which bead is in which well. This is a 12-plex respiratory panel that we developed under the DARPA program, just showing the kind of data that we can generate here, this endemic coronavirus and influenza A that we're looking at here. So roughly half of these data points are in the digital domain, and roughly half are in the analog or qPCR domain. So some nucleic acid, some RNA experiments. This is showing this ability to turn off nonspecific binding, right? So we call it noise canceling. So we're looking at 6 different cytokines in this particular case, and so the bar graph over on the far left, right, we're not using the noise canceling strategy. We're using primer pairs that work for all of the targets for the nucleic acid reporters in solution, and for what is it? IL-1 beta that is what we're going after. We're at what I think 100 picograms per milliliter concentration here. And so we can see that we have a lot of background signal from IL-1 alpha, right, and some of the others as well. Then as we move to the right, we've turned on our noise canceling strategy, so we have a unique forward primer that's coming from the bead. And we've greatly diminished this nonspecific background signal, and then the different colors, right? We're putting in 100 picograms per milliliter of the different targets here in this 6-plex cytokine assay. And this is just demonstrating that you can actually do a multi-omic analysis, look at nucleic acids and proteins at the same time. You have to use different workflows, right, to do the sample prep to put your proteins or nucleic acids onto the beads. But then once that processing has taken place, you can then mix those beads together and run them all on a single device. So this is what 3 cytokines and 3 nucleic acid targets that are shown in this data here. So it's a little marketing oriented here, but Cadeta's launched a product. So Cadeta means little tail in Latin, right? So we got that from the little tails of our wells that we have. But Cadeta, right, is also, right, a musical term, a little ending to a musical piece. So we have this musical sort of theme here. So we call this the concerto system. It includes an instrument that is a reader and chips and reagents. And the chips have 8 lanes, so it can run 8 samples at a time, 250,000 PCR wells per lane, so 2 million experiments that you could form on one of these chips. And has sort of some cute little features of all the sample waste actually goes into the sponge that is up at the top. So the device is sort of self-contained that you can pitch. Okay, so we're shooting to deliver 20-plex assays for both nucleic acids and proteins here when we launch this system. State-of-the-art sort of sensitivity, sub-picogram per milliliter for proteins, and targeting single-digit copies per microliter for nucleic acid assays. Okay, so I'm running out of time, which is a good thing because I'm also out of slides. I could try to tell you that I did all this work myself, but you wouldn't believe that, of course. And so it's, it's all these people who've wandered through my lab over the years that have generated this data and obviously intellectually contributed a great deal as well. And I don't have a slide with all the people at the companies that actually, produced some of our technology and IP into real products, but there's a long list of people there as well. And of course, to have people, you need money, and these are some of the sponsors that we've had in these areas over the years. So I just want to finish up with something that it's a quote from Jack Haldane, who's a famous biologist from the UK. Going back, as you see, 1963 is when this quote came out. So I'm paraphrasing, but when you have a new technology or a new idea, right, that the first sort of response from the community is that it'll never work. And I have lots of examples in microfluidics as well as miniature mass spec where people told me, this will never go anywhere. In fact, in microfluidics, I visited a lab of a well-known company where my host at the end of the day told me I should go home and keep doing spectroscopy. So it'll never work. And then, it works, but it's unimportant. It really doesn't solve any problems. And then sort of the next phase is that, okay, maybe there's some niche applications here that we could use this technology. And then the last one was it was a no-brainer from the start. And I actually, in the case of microfluidics, I had someone actually tell me that exact statement. Oh, this was a no-brainer, which I agreed with, of course. So anyhow, thank you for your attention. Appreciate it.
Thank you so much, Professor Ramsey, for such a great talk. We accept all the questions. We have time. Please go ahead. Please introduce yourself and your affiliation, please.
I have a question about the index migration time. You mentioned that significantly improved the precision, like decrease from 4% to 0.5%. Could you elaborate a little bit more how you apply this index migration time?
Yeah, I'd say it's, we didn't invent this at all. It's sort of a standard technique that dates back to the 50s, but you basically, you could use like one marker, one molecule, right, and basically divide its migration time into the migration of all your other peaks, right? So as it moves around, right, it should correct for the variation migration time of your sample components.
So it's similar to what we use like an internal standard?
Yeah, for migration. It turns out that just using one marker, which is a good start, isn't very effective, that we're using more than a dozen markers that span the migration time of our samples, right? And so that does a much better job than just one internal standard, if you will.
Thank you.
So I'm Hermann Wetzig from the University of Braunschweig in Germany. Impressive, Mike, really, what you showed. And what is your vision of the future? Would you rather think that it's mainly, as you also showed, process analysis and parallel work, just using the speed, or would you also think that in some cases it would be nice to have these small probes to explore, maybe outer space, or maybe some places which are now rather easier accessible by drone, something like that? So for geological studies, something like that? What do you think?
Well, that's possible. Actually, we did do some work for NASA of doing CE and zero gravity. Yeah, Chris Culbertson, who was a staff member with me at Oak Ridge, right? He got to fly a CE rig on the Vomit Comet. It's the airplane, right, that dives and gives you, 20 seconds of zero G or something. So, yeah, there's application space exploration. Rich Matthews spent a fair amount of time, right, developing, separations, right, to look at D and L amino acids, right, in Mars or wherever, right, to try to discover the origins of life. I haven't heard anything about that for quite some time. Pardon me? Yeah, right, yeah, maybe Elon Musk will do that, but the small footprints and the small sample sizes are an advantage in many cases. I should also point out that it's a disadvantage sometimes as well in terms of looking for the needle in the haystack sort of types of samples or targets, right, where the nanoliter sort of injection volumes that we use aren't the best. And there are ways around that. We've implemented. I didn't talk about it. I know Kari probably talked about it yesterday, using transient ITP in many of these experiments to inject larger volumes and concentrate them to improve detection limits while maintaining separative performance. But in terms of predicting the future, that's always a tough one. I didn't bring my crystal ball this morning, but I think there's always lots of room for progress here. It's not a very good answer, maybe, but
thank you very much.
Yeah, sure.
So thank you, Mike, as always, very impressive. But I also have, some of these hiccups on some ideas that I've been wondering about for years. You showed ELISA. Bioassays, I think, is a huge pain in the ass of a lot of people here, perhaps, or not them personally, but then at least their companies. And I'm always thinking, if I see ELISAs, like it's too complex. Why can't we simplify this? And what can we take the concept of one of the binding steps in ELISA into the CE and especially the CMS zip chip, so we avoid all these things? Now, I can appreciate the multiplication and the sensitivity step, but do we in all situations need to be so complex, or can we combine the concepts and from affinity, capillary electrophoresis, zip chip, mass spec to something like that?
Jay, I don't know. ELISA types of experiments, they're complicated. We're trying at Cadetta, trying to make them simpler. There's still, still a workflow, a sample prep associated, which is not trivial. It's not like the zip chip of, dilute and shoot, right? And when it comes to proteins and mass spectrometry, right, I always have trouble. I guess it depends on what you're trying to do, but doing protein analysis with mass spectrometry, right, if you're looking for post-translational modifications, variations, right, you're not going to do that very effectively with ELISA. But, doing a femtogram per milliliter sort of detection of a protein, you're not going to do that with a mass spectrometer, separation in a mass spectrometer either. So I don't know what. If I had a really good idea, I probably wouldn't tell you.
We can talk later.
Yeah, there's some interesting affinity types of experiments that have been done by CE over the years, right? And I don't know, maybe you know, that I don't know of any that have really gotten traction. But.
Well, the point is, we usually need some affinity step in potency assays, which is part of the bioassays. And of course, ELISA sometimes fulfills that requirement, but it always makes me itch. Being a separation scientist by heart, so and the feeling that every step that complicates our analysis also adds on to uncertainty. And if we want to develop something that is applicable in a QC manufacturing environment, it has to be simple. That's kind of, and you have this philosophy as well, so I was kind of thinking, like, can we make a nice marriage there?
Yeah, I mean, that was the attempt with Rebel and ZipChip, right, to make sample processing simple and limited hands-on time. But I mean, I don't know. I'd be interested if anybody has ideas of where, you could use sort of the Cadetta technology I talked about in biomanufacturing. We've talked to people some about that, and it's sort of like looking at host cell proteins. Our sensitivity is too high, really, to go after those. But anyhow, I'm going to be here all week, so anyhow, I'd be happy to talk to people if you have ideas or questions beyond this morning. So. Thanks.
Hey Mike, this is Ruchi Rustandi from Merck & Company. So I would say that very thank you for the presentation, your great overview, and I think people in industry in general thank from your lab, make our life easier with the development of obviously call it the blue box, and we call it in the time is the caliper technology, and then you have 908 device. So nothing's perfect, but overall it's a great technology, and we appreciate for your lab. Of course, you're coming up with the two technology coming in, and hopefully you'll be successful that well as the other two. The other one I want to ask you actually a technical question a little bit. So I didn't get actually this is for the Codiva. Is that what you call Codiva? So when you do the ELISA part, how do you actually get rid of all those non-specific binding again? Is this like software subtraction, or how do you do that?
Let's see. I didn't quite get the last sentence.
So when you do a non-specific binding, so how do you get rid of the non-specific binding? Is that subtraction from the software, or?
No. So it's done with the biochemistry, right? So these beads, right, are pulling the sample into a well, an isolated well that actually has a volume of 100 femtoliters, right? So it's one molecule in that well is like picomolar concentration. But anyhow, if the wrong detection antibody arrives in that well, right, there won't be the appropriate primers for amplifying the reporter nucleic acid on the secondary antibody. Right, so there's no software involved at all. It's basically, DNA hybridization, right, that is taking care of canceling the signal.
That's good. So one lasting question is, I remember that this technology, detecting ELISA using the primer has been done. I don't remember, it's a new company that I think remember they detect with the whole cell protein. Is that something you're familiar with? I think they apply to detect whole cell protein using similar technology. Is that correct or no?
Immuno-PCR, right, is not new, right? David Baltimore's lab did that, 20 years ago or so, using well plates. So I didn't want to imply that immuno-PCR was invented by us, right? It's the noise canceling that we invented. But I'm not familiar with the company, but.
All right.
Yeah.
Thank you.
Yeah, sure. Exact time.
Yes. Any other questions? If not, thank you so much again, Professor Ramsey, and please join me in welcoming. We are doing great on time. So now we come to the very special moment in CE Pharm Symposium, which is a CE Pharm Award presentation. The legacy of CE Pharm Award started back in 2006, and the first awardee was Roberto Guzman, and the most recent one, 2024, was Bob Kennedy. And in between, we had such a pile of great scientists. We are lucky that we have, I think, 5 of them here. So I would like to invite them to stand up. Please, Tim, Gary, David, Jetchi, Herman, and please join me in giving them a round of applause. Thank you so much. We love you. So, CE Pharm Award 2025. I'll try to make it short and easy. You can predict. So, the awardee is recognized for pioneering advancements in capillary electrophoresis and its applications in pharmaceutical analysis. So specific. Contributed to the development of innovative separation techniques that enhance precision and efficiency. Demonstrated leadership in bridging academia, industry, and regulatory expectations for CE methods. Mentored the next generation of scientists and expanded the global impact of CE technologies. Getting close. Played a transformative role in advancing microfluidics and miniaturizing CE systems. Pioneered the methods that revolutionized high throughput and portable analytical testing. The awardee has more than 300 peer-reviewed publications and more than 2010 issued patents and non-specific number for pending ones. I bet the number increases day to day. The awardee developed technologies that became the foundation for lab-on-chip and next-generation analytical problems. I think we can guess now. So this year, the CE Pharm Award proudly recognizes Mike Ramsey, University of North Carolina, Chapel Hill. Mike, please join us here. This one. Okay. Pardon me, just I'm going to give a few words before the pictures. Yes, please.
Well, yeah, thank you. That's quite the surprise. I was sort of curious. I know I'm familiar with sort of the secretive nature of determining who the award winner is, and so I was curious who it might be. I actually didn't have a clue. So you did a really good job keeping it a secret. So anyhow, yeah, thank you to CASS and the selection committee and all of you for being here today. Thank you very much.
Thanks, Mike.